Hey guys, I just wanted to make a quick video over what we've covered the last couple of days in class. I had a little change of plans. I was originally we were going to start off the semester with genetics, but I decided to go ahead and because it's in the news, we hear a lot about it on TV and radio about the vaccine for the COVID. So I thought I would go ahead and talk about protein synthesis. And in the process of talking about protein synthesis, I would kind of incorporate in a little bit about the COVID-19 vaccine because they kind of go hand in hand. So let's go ahead and jump into this. First of all, protein synthesis is basically the way we go from our DNA, which is our genetic code, into flesh and blood, basically protein, which is what you're made of, your skin, your nails, your hair, your muscles, and all that. I like to use an analogy when I talk about this of building a house. So when you build a house, the first thing you have to have is a blueprint. And when you have that blueprint, you don't typically give that original blueprint out to the plumber and then get it back and then give it to the electrician and then get it back and then give it to the heat and air people and then get it back and then give it to the framers. And if you did that, there's a possibility that your blueprint could get damaged, might get torn, might get coffee spilled on it, could get muddy, or it could get dirty. And by the time you got it back, in the end, it probably might be pretty unreadable. So typically what you do is you take that original blueprint and you make copies of it. And you give the plumber his copy, and you give the electrician his copy, and the framers their copy. And they can use their copy to build your house. And that's kind of the way protein synthesis is with us. Your DNA is like that original blueprint. We keep it safe and secure inside the nucleus in your cells, and it never leaves there. And so what we do is we make copies of that DNA in the form of RNA, which is more specifically a messenger RNA. And then we send that RNA out of the nucleus, and it goes to a ribosome, and the ribosome reads it and makes a protein from it. And that way our DNA gets protection and it never gets damaged, and the RNA will go out and it will serve its purpose, but it eventually degrades and gets broken up and recycled, and more RNA will get made inside the nucleus. So that kind of is a good analogy I like to use. So let's go ahead and get into this. So there's two steps to protein synthesis. The first one is called transcription. The second one is called translation. Now, an easy way to remember those two: transcription has a C in it, translation has an L in it. C comes before L in the alphabet, so that helps me remember that transcription always occurs first. And really, this idea of transcription, if you transcribe something, you're just copying it. And that's really all we're going to be doing in the first step. So in transcription, what we're going to do, DNA is used as a template to make a copy of mRNA, or that stands for messenger RNA. Now, before we get into RNA too much, I want to talk about some differences between RNA and DNA. First of all, RNA is single-stranded as opposed to the double-stranded DNA. The sugar is different. DNA actually stands for deoxyribonucleic acid, where RNA is just ribonucleic acid. So the sugar in DNA is deoxyribose, whereas in RNA it's just ribose. It's not a big deal there. That's not going to really affect anything that we're going to be learning about today. But the last difference is probably the biggest one that we can talk about is this: there's no thymines in uracil. So there's going to be I'm sorry, I said that backwards. There's no thymine in RNA. Instead, there is uracil. Okay, so, and I'll get into that more in just a little bit. So, that analogy I gave you earlier, so the blueprint copies that you make are to the blueprint as the mRNA is to DNA. And then to take that one more step, the blueprint is to a house as the DNA is to you. Before we get into that, into translation, I wanted to make sure, I didn't have this in the notes anywhere, I did it on the board, but I wanted to make sure you remembered that when we're talking about DNA, remember there's 4 typical letters that we will use in DNA. Let's see if I can write on the board, on the screen here. So you remember there are A's, which stands for adenine, and they bond to T's, which stands for thymine. And then C's bond to, which is cytosine, will bond to G's, which stands for guanine. However, as I mentioned just a little bit ago, in RNA there are no T's. So this is in DNA. So write this here, DNA. So in RNA, hope you can read my writing. I'm writing this with a mouse. RNA, U's will take the place of T's, so I kind of like to do this. We'll circle that T and draw a line through it. There are no T's. Instead, A will bond to a U, okay, which stands for uracil. And so what that means is if you have an A in a DNA sequence, that corresponding messenger RNA is going to be a U instead of a T. Now, if you have a T in the sequence of DNA, when it makes that copy, when that base pairing pairs up, you can still have A's in RNA, but you just can't have any T's. So if you have a T, you can still have an A, but if you have an A, you're going to have a U instead of a T. I hope that makes sense. Let me know if it's confusing to you. So let's go ahead and move on here. So the second step in protein synthesis is called translation. So when we talk about translation, we're going to translate that code into an amino acid sequence. And eventually that amino acid sequence, that string of amino acids, will become a protein. So let's talk about, so that messenger RNA that you made in the transcription is going to leave the nucleus, and it's going to travel to a ribosome. And if you remember way back in the fall, we talked about ribosomes are the site of protein synthesis. So at that ribosome, it's going to translate that mRNA into a sequence of amino acids. And it will read that messenger RNA 3 letters at a time. Those 3 letters at a time are what we call a codon. Okay, so a codon are 3 letters that correspond to a specific amino acid. Now, there's this chart that I will provide for you on any worksheet that we do or on the test, but I wanted you to be familiar with it. This is the kind of the translation sheet. So this is going to give us a list of codons and the amino acids that they code for. So the way I like to tell people, tell students, is that each codon codes for a specific amino acid. However, an amino acid can have more than one codon code for it. So for example, if we were to look at AAA, that codes for lysine. But you'll notice that lysine can have 2 different codons that code for it, AAA and AAG. ARG, arginine, has actually 6 codons that code for it. So the way these codons in this chart works, the first letter is here, and the second letter is here, and then the third letter is over here. So if you had U, C, and then A would be UCA right there. It codes for serine. Okay, so these letters are your codons. These are part of your RNA sequences. So you're going to have A's, U's, G's, and C's, and then these others, these are abbreviations for amino acids. You don't have to memorize this chart. You don't have to memorize the amino acids. I'll provide it for you, but these are just abbreviations for the different amino acids. So let's move on here. This is what's called a transfer RNA. So transfer RNAs are what bring the amino acid in. You'll notice on one end has an amino acid, and the other end has what we call an anticodon. So the anticodon will base pair to the actual codon. So if the codon reads GCC, the anticodon will be CGG. And that specific transfer RNA, if we looked at that chart, GCC is going to code for, let's find it here, GCC is right here is alanine. So that specific codon is going to code for that amino acid would be alanine. little bit more about transfer RNA. It says a transfer RNA with an anticodon carries amino acids to the mRNA. And again, that anticodon is actually found on one end of that tRNA, and there's 3 letters that match up to the codon. And again, that transfer RNA, the tRNA, is going to have an amino acid on the other end. What does this have to do with COVID vaccine? The COVID vaccine is a new vaccine in that it's an mRNA vaccine. So we just learned about what mRNA is. mRNA is a copy of your DNA. So what they have done with this vaccine, as opposed to other vaccines, other typical vaccines may have a weakened form of a virus or a virus that's non-functional that won't actually reproduce inside of you. But this one does not even, it just has a small little segment of that virus's RNA that in this one it's coding for the little protein spikes that you see on the surface of the COVID-19. And those little protein spikes are what really allow that virus to get into your cells. So it's just so what happens is your cell will take in that little tiny segment of that RNA strand, and it will read it, and it will make the protein spike. That's all it will make. It won't make the whole COVID. So you cannot get COVID-19 from this vaccine. It only makes the little spikes that are on the surface. And then your body will your immune system will recognize that spike, and it will make antibodies for it and basically signal it for destruction. And then it will remember what it looks like. And if you ever get if you actually get a COVID virus inside you, that your immunity should recognize it and say, I've seen that spike before, and attack it and kill it. And so that's how this one works. It's pretty cool. This is a new technology that's been around for about the last 30 years. They've done a lot of research, but this is the first time we've actually used it on a widespread vaccination of the public. So so far, really the main concern, it seems like, with it is some people are having some allergic reactions, but those are pretty far and few between. And those are primarily probably people that are having allergic responses to different other things in their life, maybe food or bee stings and things like that, or wasps. And so those, sometimes if you have a lot of allergies, you may have an allergic reaction to this vaccine. But, you could always just check with your doctor to see what they recommend. But anyway, but it has a very high success rate. The first round is like 50% coverage, and then the second round, which is usually around 28, 30 days later. Some cases, I think, depending on the vaccine, might be a little less than that, but it raises up to about 95% protection. So it has a really good success rate, and hopefully it takes care of this COVID for us. If you want to get some more information on protein synthesis, I've gone through this pretty quickly, but check out one of the Amoeba Sister videos, or go on YouTube and search protein synthesis, and you'll see other videos that might actually show the process more detail. But it's really not too complicated of a process. You're basically taking your DNA, transcribing it to RNA, and then translating that RNA into an amino acid sequence. So it's not too complicated. We'll probably have some type of activity sheet or something the next day or two to get some practice in that transcribing and translation with it. So be looking for that. See you guys later. Bye.